Today I'm going to talk about DNB-seq Cool MPS and how it could enable new discoveries in cancer research. And I should note that this work is being done in Nick Matthews' lab at the Institute of Cancer Research in London. So DNB-seq is our unique technology. What is it? The first step is the DNA nanoball. Like any library prep you're probably used to, DNA is ligated to adapter sequences, except for us, we circularize it to form a single-stranded circular DNA, or ss-circ DNA. This is to facilitate the rolling circle replication, or RCR, to generate the DNA nanoball, or DNB. Our patterned array is specific to us. It's designed to trap DNBs onto spots. Finally, our chemistry is Cool MPS, which consists of unlabeled block DNTPs and labeled base-specific and block-dependent antibodies. They enter the flow cell and are extended by multiple strand displacement amplification, or MGI. The bound antibodies are excited by green and red lasers, and the emission is filtered. So this is what our ss-circ DNA looks like. And this is the splint oligo. And this is the Phe29 that carries out the rolling circle replication and exhibits the strand displacement activity. So essentially this just rolls around this circle until we stop the reaction. And when we do, we get the DNB. So the libraries are in this controlled enzymatic reaction generate this long continuous single strand. So it's the unique sequence, the insert, adapter, sequence, barcode, etc. And one copy converts to about 300 to 500 copies. So the ss-circ DNA libraries will vary in size just like any adapter ligated fragment. So if you have your smallest insert will give you the smallest circle. Your largest insert will give you the largest circle. And this medium one will give you your medium circle. The ss-circ DNA libraries are converted all in the same reaction, and that reaction is stopped after 25 minutes. Each circle results in a DNB of the same size, but with a different number of repeats. And as I said, the copy ranges from 300 to 500. The smallest fragment size will have the most repeats on the DNB. So the smallest one will be 500, the largest one will be 300, and then the medium-sized one will be about 400. But they're all the same size DNB. So there's no preferential loading of small fragments because there are no small fragments. So the diversity of the sample is preserved, and that leads to higher mapping rates and rarer sequence being represented. Cool MPS is our innovative method. So in standard chemistry, cleavage of the fluorescent moiety from the fluorescent DNTP on the read, it leaves behind a ligand that physically inhibits the next DNTP from binding. This is called a scar. The buildup of scars over the read causes a drop in quality. So for a paired N100, you would see 2 drops in quality. So you'll see that it starts out high and it drops over the course of the first read, and then it bumps up and then it drops over the course of the second read. Now in Cool MPS, we use nucleotides with a natural base and an extension block rather than fluorescent DNTPs. And we also use labeled antibodies that are base-specific and block-dependent. The antibodies bind specifically to just one base. So let's look at what happens when you switch out the standard with the Cool. That's what happens. So there's also another interesting difference, and that is that with standard chemistry using fluorescent DNTPs, there is a quenching of fluorophores. We do not see this quenching on the fluorescent antibodies. So not only are there no scars, there's no quenching of the fluorophore. So that means that the antibodies if we have efficient washing, there's very specific binding, and that culminates into having really high signal-to-noise ratios, low background, and that means higher confidence base calls. And it also means that this is sustained over the course of the entire reaction. So how does this work? You start with the DNB template and the sequencing primer. You introduce the unlabeled nucleotides and the base pair, and then we introduce the fluorescent antibodies, and then that pairs. We wash the excess antibody away, image, and then we remove and wash off the block and the antibody, and that leaves a regenerated natural nucleotide without scar, and then repeat. Overall, this means high signal-to-noise ratio, and that means higher accuracy base calls. There's no scarring of bases, so that means longer reads without a drop in quality. There's the added advantage that the antibody industry supports biological drugs, so it's actually a cost-effective choice. This is the G400. This is the machine that we have at ICR. This is our most flexible instrument. It's our mid-sized one. And it has two sides that are independent of each other. So at ICR, we used 4 whole genome from mouse cells prepared with PCR-free library prep. And we compared cool MPS with standard chemistry. The quality distribution, the Q30 to Q40 scores versus the cycles in the raw FASTQ files look like this. So this is the standard chemistry, and this is when we substituted the exact same run, same libraries, exactly the same in every way except cool MPS. That's what happened. So you'll note that on the first read, there is practically no drop. The only drop, and it's a small drop, is only on the second read, which made us really think, what if we didn't have a second strand? What if we just carried on with a single end? And then, also, the summaries of the FASTQ statistics for standard versus cool chemistry showed low error rate and a shift to higher quality estimation. So this is the standard chemistry, and you'll note that the Q30s are really good, actually. Very low error, low N. But this is substituting cool MPS. So it goes to just incredible Q30s, incredibly low error, and no N. And this is just a little bit of bump in the depth. So it goes from this depth to that depth, just switching to cool MPS. There was almost no per base N content for the cool MPS versus standard. So this is the N for standard, and this is for cool MPS. There was no overrepresented sequences and adapter content. Better percentage mapped reads, more reads, and higher coverage using cool MPS. So the thing to note here is really, I think, the median and the mean, the depth and coverage. So you'll see that it's much tighter, around 30 here. Higher mapped reads, more reads. The duplicates aren't really going to be a thing because they're PCR free. So changing only the chemistry to cool MPS improved the Q30 scores and reduced errors, yielding more data. This was the first reported outside of MGI and is from the first cool MPS sequencing run we ever did. The data demonstrates excellent performance in mapped reads, coverage, and error rate. Cool MPS technology has opened the door to not just unprecedented quality reads, but also quality sustained over a long read. Further work is indicated for applications with demanding sensitivity and longer reads. So these are the two applications. A collaboration with UCL Cancer Institute using their novel method of sequencing methylation in circulating tumor DNA, and long-read sequencing of the variable regions in immunoseq. So, of course, this is very important work doing TCR repertoire profiling, and we're looking at RNA and also germline. Thank you very much.